Welcome to this video on the primary identification of bacteria. Once a pure culture is obtained, a few basic tests can help identify the bacterium to the generic level. Primary identification includes simple and rapid tests such as the Gram stain, McConkey-Agar growth, Catalase, Oxidase, Motility, and oxidation fermentation tests. First, we will learn about Gram staining. The Gram stain establishes whether the bacterium is Gram-positive or Gram-negative and also reveals morphology, whether the cells are cocci or rods. Certain species may show partial decolorization. Endospores appear as clear areas inside cells and may indicate genera like Bacillus or Clostridium. When Gram stain results are equivocal, alternative tests like the Lana test, potassium hydroxide test, and vancomycin susceptibility help differentiate between Gram-positive and Gram-negative organisms. First, we will learn about Lana test. Next is the Lana test, which helps differentiate Gram-negative from Gram-positive bacteria. To perform the test, take a sterile swab that has been impregnated with L-alanine 4-nitroanilide. Touch the swab gently to a single colony from the culture. If the organism is Gram-negative, the swab will turn yellow due to enzymatic cleavage of the substrate. If there is no color change, the organism is considered Gram-positive. Next, potassium hydroxide test. In this test, Gram-negative bacteria lyse and produce a viscous gel within 60 seconds, while Gram-positive bacteria do not. Next, vancomycin susceptibility. Most Gram-positive species are susceptible to vancomycin, whereas Gram-negative bacteria are uniformly resistant. Bacteria are classified as cocci or rods. Pleomorphic bacteria may look cockle but usually show rod forms as well. Listeria also shows cockle forms in young cultures. Growth on McConkey-Agar. McConkey-Agar is used to check for growth of Gram-negative bacteria. It inhibits most Gram-positive organisms and supports Enterobacteriaceae. However, different commercial versions vary in ingredients. Catalase test. Catalase is an enzyme that breaks hydrogen peroxide into water and oxygen. This test detects the presence of that enzyme. The reagent used is 3% hydrogen peroxide, which must be stored at 4 degrees Celsius in a dark bottle. When working with bacteria grown on blood agar, care must be taken because red blood cells naturally contain catalase and can produce a false positive reaction. In method 1, a small amount of bacterial growth is taken from the top of the colony, to avoid the blood agar, and placed on a clean slide. A drop of hydrogen peroxide is added. Immediate bubbling indicates a positive test. In method 2, hydrogen peroxide is added directly to the colony on the plate and also to an empty area of the agar. Some bubbling will occur from the blood agar, but significantly greater bubbling on the colony indicates a positive reaction. Next, oxidase test. Oxidase test identifies bacteria that possess cytochrome C oxidase. Anaerobic bacteria are oxidase negative. The reagents used must remain colorless. If they turn dark blue, they are oxidized and should not be used. Autooxidation can be slowed by adding 1% ascorbic acid. Colonies should be taken from non-selective media that do not contain glucose or nitrate. Only a glass rod, platinum loop, or plastic loop should be used, because nichrome loops can produce false positives. Pseudomonas aeruginosa is used as a positive control. In method 1, a filter paper is moistened with a 1% solution of tetramethyl p-phenylenediamine. The bacterium is streaked across it. A dark purple color appearing within 10 seconds indicates a positive reaction. In method 2, equal volumes of 1% alpha-naphthol in ethanol and 1% p-aminodimethylaniline oxalate are mixed. A drop is placed on the colonies. A blue color appearing within 10 to 30 seconds indicates a positive result. Weak reactions after 2 minutes are ignored. Commercial oxidase strips or sticks can also be used according to manufacturer's instructions. Motility tests. Most bacteria are motile due to the presence of flagella. Motility may depend on temperature, with some bacteria motile at room temperature but non-motile at 37 degrees Celsius. In the direct microscopy method, a young broth culture, 2 to 4 hours old, is prepared at room temperature. A hanging drop preparation is made by placing a drop of broth on a cover slip and inverting it over a ring on a slide. Under the microscope, true motility is indicated by directional movement, while Brownian motion appears as random vibration. If the organism appears non-motile, results should be confirmed using motility media. In the semi-solid motility medium method, 2 tubes are stab inoculated and incubated, one at room temperature and one at 37 degrees Celsius. If tetrazolium salts such as TTC are added, they are reduced by bacterial growth to form a red pigment called formazine. Motile bacteria produce diffuse red or turbid growth throughout the tube, while non-motile bacteria grow only along the stab line. Tubes are examined at 24 and 48 hours. SIM medium can also be used to detect motility, as well as indole and hydrogen sulfide. Last one, OF test. The oxidation fermentation test determines whether a bacterium metabolizes carbohydrates oxidatively or fermentatively. The medium contains glucose and bromothymol blue as a pH indicator. The medium is green at pH 7.1. When acid is produced, it turns yellow at pH 6.0. Before inoculation, the medium is heated in boiling water to drive off oxygen and then cooled. 2 tubes are stab inoculated. One tube is sealed with a layer of sterile paraffin oil to create anaerobic conditions, while the other is left open. The tubes are incubated at 37 degrees Celsius and checked after 24 hours and then daily for up to 14 days. If both tubes turn yellow, the bacterium is fermentative. If only the open tube becomes yellow, the organism is oxidative. If no change occurs, the bacterium may be unable to grow in the medium or cannot utilize glucose. For fastidious bacteria, the medium can be enriched with serum or yeast extract. For Staphylococci and Micrococci, an alternate OF medium containing tryptone, yeast extract, agar, and bromocresol purple is used. After autoclaving and cooling, 1% glucose and 1% mannitol are added. Bromocresol purple transitions from purple at pH 6.8 to yellow at pH 5.2 to indicate acid production.